All right, hey everybody, Mr. Boyd here, marine biology, phylum Chordata, Haeckel's embryos, getting a sense for this kind of bridge that we're dealing with and how we think about invertebrates and vertebrates on a couple of strange taxonomic decisions and taxonomic possibly conundrums here. So we're exploring that a little bit. The goal being to get a better handle on these creatures, to understand them as their characteristics dictate. And there's always going to be a tension there between theory and evidence. And that's something that takes a while to even get close to resolving or reconciling. There's always going to be that tension where a prevailing theory has evidence that doesn't support it. And one of the questions we need to be asking as scholars, as Christians who value our understanding of general revelation via the scientific process, is when does the evidence reach a place where it actually should cause the demise of or decline of a certain theory. So theories really they're big picture. They take decades to develop. They tend to be a lot more creative and maybe even kind of apart from evidence at times. And so the scientific theory is a challenging concept. It doesn't always comport with evidence. But that doesn't mean we should throw them all out either. It doesn't mean that just because we have some lines of evidence that don't support a theory, that doesn't get rid of the theory's value. I think the best example of a scientific theory that's kind of changed and been refined and withstood the test of time is plate tectonics theory in continental drift. Alfred Wegener, continental drift. He didn't have a mechanism to explain what he saw as just really clear lines of evidence supporting that the continents were once connected, fossils that are similar along coastlines that would have once been adjacent to each other, the shapes of the coastlines fitting with each other, the strata of the geology that is very comparable on both sides of the Atlantic Ocean, for example, if we think about the east coast of South America and the west coast of Africa. And so this theory has grown and been refined, and really, I would say, stands as an excellent example of what you hope happens when a theory is being subjected to evidence. And so, who knows what will happen with evolutionary theory? But we're in that realm right now, and we're talking about a really important book that I introduced to you. I just mentioned it last week. It's up in ThinkWave today. It's called Icons of Evolution. I was first exposed to it 15, 20 years ago. I was able to actually bring the speaker here to where we are locally and had him be a headliner for a homeschool science conference that we put on. And so you're going to read the introduction to his book tonight. And then tomorrow night, you're going to be reading the first or the fifth chapter regarding Haeckel's embryos. We introduced that icon. We've introduced Haeckel a little bit and what his depiction of various vertebrate classes in the embryonic stage, what he's attempting to accomplish there. And so this is going to pick up where that left off. The introduction is going to be more about the science process, thinking about important definitions, et cetera. And then the chapter is going to be, there's a lot of detail in there. So it's going to take some wading through. There's a lot of history. It might seem a little bit redundant at times, but I want to make sure we leave with a really strong sense for what's wrong with this picture or depiction that Haeckel put forward, along with lots of other folks. Okay, well, let's jump into the topic of the day. We want to get through all three subphyla, generally speaking, basic characteristics, what they have in common, what's different about them between the groups. And at the end of the day, I want us to be able to think clearly about, should these three subphyla all coexist under phylum Chordata? Does that make sense morphologically? Does it make sense? We're not getting into molecular DNA analysis at this point. We're just doing gross morphology. But at least from that perspective, should these three stand distinct from each other? Should they be in three separate phyla, maybe, or two, or does this configuration make the most sense for the biologist? All right, here we go. All right, there we go. This is what we saw last week, and there's our. We talked about this phrase ontogeny recapitulates phylogeny, quite a mouthful, and I had you guys dissect those three words. Recapitulates means walking through the steps of a process again, right? Or restating something in a more intentional and kind of thorough way. Ontogeny was coined by Haeckel, and that meant embryological development. So ontogeny is the embryological development process. It happens for all vertebrates. It happens in utero for live creatures that give live birth. It happens in the amniotic egg for creatures that lay these eggs that have a lot of similarities. They're removed from a womb, removed from the body, but the structure, the membranes, lots of what's going on inside an amniotic egg is very similar to what's going on inside a creature that's developing in utero. So that's ontogeny, the development from a zygote, sperm and egg fuse, zygote, all the way to hatching or birth. That's ontogeny, or embryological development. And phylogeny, again, is evolutionary history of that creature. And we looked at several different hypotheses in biology class, if you remember. Evolutionary tree of life, tangled briar patch, and then creationist orchard as diagrammatic hypotheses of what might have occurred leading up to the diversification of all living things that we see today. So that's phylogeny. And so Haeckel's phrase here, Haeckel's theory, which was also called biogenetic theory, is that as you look at any of these classes of creatures, we have the fish on the far left, then a salamander, then a tortoise, then a chick, then four different mammals. And I always mix them up here. We have a pig, we have a cow, and I can't remember the other one. And then we have, of course, a human here. And these are kind of handpicked to put into three different stages of development. We know that's a gross understatement of all the development that's actually occurring. But his idea, again, is that as I went through ontogeny, embryological development from stage 1 to stage 2 to stage 3, if we're to look at all those stages, we would see the adult forms of our ancestors, meaning we'd see a lower mammal. At some point, we'd see a bird, a reptile, an amphibian, and a fish at various points. And he used this to say that this supports a macroevolutionary theory, a descent with modification from a common ancestor, right? So that's the gist. To review that, we're going to be looking at all of these different groups of creatures here eventually in the marine biology textbook. And in our textbook, we're straddling module 5 and going into 6. And we start talking about the fishes, the three classes of fishes, Agnathochondrichthyes and Osteichthyes. And those are, of course, the jawless fishes like the hagfish and lamprey, cartilaginous fishes, chondrichthyes, sharks, rays, skates, and others. And then the osteichthyes are bony fishes. Amphibia, reptile. I had a big misspeak last time regarding marine reptiles. Meant to say that there's just one marine lizard, but lots of marine reptiles. I've done a little more homework recently to find out that there is one species of frog that does spend a significant amount of time in brackish water. So water that's more saline than fresh water, but less saline than most ocean water. A couple other frog and toad species that will eat creatures from this zone and are able to get the salt out. But one frog in particular that lives in this zone and spends some significant time in that brackish water is the crab-eating frog, also called the mangrove frog. So that's new information for me after doing a little bit more homework. There, of course, we'll talk about seabirds and then mammals. All right, so goals of today: distinguishing the major characteristics of the three subphyla again, and then describing the first two fully, and then getting into vertebrates. Here we go. I have to keep an eye on my clock here. It's hiding from me. All right. So these three features, there's actually a couple more that are mentioned, but these are the ones that feature prominently. The urochordatans, cephalochordatans, and the vertebrates all have these features at some point during their life cycle. The dorsal notochord is kind of a preliminary vertebral column or preliminary backbone, dorsal tubular nerve cord, and then these things called pharyngeal pouches. A couple of other characteristics that they all have in common too that are mentioned. One is they're called a post-anal tail. So a tail that extends past the anus. That's a feature that all three of these groups have as well. Of course, we don't see that in us, but we do see that in many other mammals and lots of other vertebrates, most vertebrates even. And there's something else called an endostyle. We're not going to get too into that today, but an endostyle, important in concentrating iodine, and to which makes that available for the body to use. Some biologists think that this endostyle is a developmental stage leading to the thyroid gland. But these are the three major ones: nerve cord, notochord, and pharyngeal pouches. Pharyngeal pouches, again, often misstated as gills or gill slits. In aquatic creatures, this develops into gills often, not always. And we'll see that today. And in terrestrial creatures, sometimes developing into jaws, inner ear tonsils, and various different glands. All right, so here are three subphyla, and you must know these three subphyla. Test Thursday on echinoderms, and this chapter, or on chordates rather, at the very end of chapter 5, the beginning of chapter 6. And so in chapter 5, pages that are pertinent for echinoderm through, that's 147, page 147 all the way through 160. And then in chapter 6, really, chapter 6, you're only getting into maybe page 160, 167. So the vast majority is from the end of chapter 5. Not going to have, the test is not going to be on all the classes of fishes. We're not getting into that yet, but these three subphyla will definitely be on there. So urochordates. Now this phylum is, and sometimes called a phylum, is usually considered tunicata. Tunicate being a generic term for the sea squirts. And so this group of creatures, the notochord, they have the notochord as larvae and the nerve cord as larvae, but they both break down and absorb and aren't present in adults. These creatures are sessile as adults. They're motile as larvae. Cephalochordates, also called the lancelets. Now, the tunicates, the sea squirts, include a lot of other interesting creatures. There's over 3,000 species. We're discovering more every year or so. Cephalochordates is a very small group, only 30 to 35 species represented. So much narrower range of characteristics in the cephalochordates. They're mostly called the lancelets or amphioxus, and their notochord and nerve cord persist. So they retain them through adulthood, right? And so they have a notochord and a nerve cord as larvae. They are retained. Urochordates have a notochord and nerve cord as larvae. They are not retained. And then lastly, vertebrates, also called craniata, they have a notochord early that then develops into a vertebral column. And of course, the nerve cord is retained inside of that vertebral column, right? So we have a vertebral column with that nerve cord present, running down inside of it, protecting. So those are the major characteristics, notochord and nerve cord, we're focusing on. We'll also look at the pharyngeal pouches and their role in this in these three phyla as well, three subphyla. All right, so there is the tunicate slide. The sea squirts. So their shape is similar to a sponge, tend to be smaller, tend to be fairly small, a few inches, few centimeters in height. And these creatures have an obvious opening at the top, kind of like the osculum of a sponge, but they have a muscular siphon. And the tissue, they have true tissue unlike sponge. It's this leathery tunic that's secreted by the body. It gives them a bit of rigidity, so they're erect or upright, and they have a muscular kind of body with an in-current and excurrent siphon. So water comes in through here and then leaves through the top. So they don't have choanocytes, they don't have amoebocytes or these specialized cells to bring water in. They actually have a functional organ system to draw water in and then expel it. And they filter their food. They filter the food with their pharyngeal pouch. Again, it's not fair to call those pharyngeal pouches gill slits. Even though they may look like gills, they don't always function as gills. So there we go. The tunicata again includes the sea squirts, which are the most well-known of the group, but it also includes some really weird creatures like this salp here. This salp here that is more like a siphonophore. If you remember Portuguese man of war, these are creatures that are colonies of zooids, colonies of single cells. We see that in several types of tunicates or urochordates as well. So single cells that will differentiate and work together to become kind of a super organism. Most tunicates live in warm marine waters, not all. Most are hermaphroditic and broadcast spawners, and most adults are sessile. Not all. That, for example, this creature is not a sessile adult. These salps are larvaceans, as they're sometimes called. Here's some more. This one is a pyrosome. A friend of mine saw a whole bunch of these washed up on the coast of Oregon a couple of years ago. I was unfamiliar with them until he sent me some photos. And so a really neat group. Many of the tunicates are often called ascidians. That's synonymous. Urochordate, ascidian, and sea squirt are almost synonymous. Here's a larvation, a really gelatinous type creature. Many of these predatory. So it's kind of a diverse group all in itself. I mean, the tunicates could constitute a phylum in my mind. It just doesn't make sense that they don't constitute a phylum. Again, if we look at the characteristics of echinoderms, right, and then we look at the characteristics of arthropods, I think tunicates could stand alone as a very unique group. I think that's my current position. All right, let's look at these features. Here's the nerve cord, the dorsal hollow nerve cord running alongside of the notochord in a larval sea squirt or ascidian tunicate. Here's the what we call the pharyngeal pouch or pharyngeal basket, sometimes called, and here it's called a branchial sac. That kind of hints at this being used for respiratory purposes, but that's not what it's used for in its adult form. So you can see it's very tadpole-like. This is an actual micrograph of a larval sea squirt. And this was noticed early on that this creature in its larval form has some interesting similarities to a frog tadpole. So some kind of a superficial comparison there, but interesting to note the similarities. The spiracle is kind of a gill of sorts. We see these very prominently in skates and rays. And we don't have anything like that, of course, in the frog. But just for historical context and some comparative anatomy, just to show this image, which I think is interesting, from the early 1900s. I think this image is dated 1905. The life cycle, or rather the adult form of this creature. Here we see a sea squirt in larval form, loses its tail, kind of like a frog loses its tail, but becomes sessile. Vast majority of these sea squirts are sessile. It's going to attach itself to a hard substrate. It's going to change significantly, metamorphose to be able to become sessile, and then continue to metamorphose to this final mature form. And look at that pharyngeal pouch here. It's massive, and it's used for straining food. It has two siphons, as you can see, water in, water out, and that siphon is directing the water at the pharyngeal pouch to pull food out. There are full body systems here. There is a cardiovascular system with blood vessels that are intact with real blood. They do have this strange ability to concentrate certain different rare metals, like vanadium, for example. Vanadium is in super high concentrations in the tunic, the leathery tunic of these adult sea squirts. I don't know why that is. Some people postulate that that's for that it would be toxic to predators, maybe. These are eaten by people, sea squirts are. And we'll watch a little video possibly of that later on when we meet tomorrow, if I can remember to pull that up. And so this tunic is secreted 3 layers of tissue like many other animal creatures, right? Endoderm, ectoderm, and mesoderm. And then we have a stomach, we have reproductive organs, and we have a digestive system, right? So superficially looking like a sponge, but very different. And I think kind of arbitrarily placed with next to the vertebrates because of all of these complex body systems and the presence of that nerve and notochord, which kind of is a circular argument for proving evolution, right? This creature must have evolved into a vertebrate because it had a nerve cord and notochord when it is in the larval stage. That image turned out terrible. I'm not sure what happened there, but this is a really rough image of the process going from larva to full adult. So it becomes sessile, and then you see a pretty significant metamorphosis over time. Lastly, with the tunic being secreted. All right, cephalochordates, the lancelets. So that was Chordata, tunicata, the ascidians. Lancelets favor sandy, warm marine bottoms. And here they have a fish-like quality to them, but not they're called cephalochordata because they kind of have head-like region, more so than fins the tunicates. These are also called amphioxus, and that's where you see them most mentioned in the scientific literature, called amphioxus. So they retain that dorsal hollow nerve cord and notochord as adults. There's not really much of a metamorphosis compared with what we see in the Urochordates. The notochord, so it doesn't develop in the vertebral column. And we do see a piece pharyngeal pouch. That's also used for filter feed. So this creature likes benthic regions, separate males and females, unlike the Urochordates, which are hermaphroditic, typically broadcast spawners. And these larvae don't last too long. In fact, larval stage can't even feed themselves. And so it's a very short-lived larval stage that develops in the water column. And then they sink to the bottom and transform into lancelets, not as sessile as adult. And here's an image of one. This is saying 600 micrometers, right? So that's 0.6 millimeters in size. So these will grow a few centimeters, a few inches. They're actually harvested for food in certain tropical regions and can reach super high densities in the sandy soil as well. Okay. There's another cephalochordate lancelet. It tends to orient itself this way, kind of like a clam, with its mouth parts near the surface of the substrate so it can grab food particles, and very reminiscent of our friends, certain Darians, or even certain polychaete worms with these tentacled or ciliated mouth parts that grab its plankton. And here we see the pharyngeal slits. That is for trapping food, which is kind of like what we see in clams. The clams are gilled, and they also those gills functions as a food trapping device as well. So that's an interesting similarity. You can see that they have a complete digestive system, 2 openings, and there's that what we call that post-anal tail, which you could see in the larval tunicate as well. This is the lancelets. Interesting organism. Here's a close-up look. Yeah, 25, 30, 35 species. A closer look. This image calls these gill slits, which is not necessarily accurate here. So the notochord and nerve cord side by side in this image, which is interesting. Ciliated mouth parts. Okay, And last but not least, over to our friends, the vertebrates or craniata. I think I called them craniata earlier. Make sure the right slide set here. Yeah, craniata. So lots of differences here. So we have the lancelets, phylum, Cephalocordata, chordata. We have all the tunicate varieties, and then we have this phylum vertebrata. So I leave it to you to determine, should these three subphyla coexist? Look at the differences we see here now. We see a complete endoskeleton, which we've had varieties of endoskeleton before, if you think about it. In the echinoderms, the sand dollars or sea urchins or sea stars, there's some significant calcareous portions of the body that are internal, but we don't see the degree of, I would say, we don't see the degree of complexity that we might see in an arthropod's exoskeleton. Arthropod's exoskeleton, jointed and functioning more like what we think of as a true skeleton. And we see that, of course, with us and other vertebrates, joints and these bones that are connected by ligaments and tendons to muscles and to other bones. Chambered heart. This is something that we see in smaller hearts, or maybe even two-chambered hearts in certain invertebrates, but this is where we see the heart really become a remarkably sophisticated organ at separating oxygenated blood from deoxygenated blood. We have a closed circulatory system and a complete nervous system, including the central nervous system, that dorsal nerve cord inside the vertebral column connected to a brain, and a fully developed cephalic region, a head region. And so bone is pretty amazing tissue. Just a reminder of what bones look like. Let me check my clock here really quickly. There we go. Good still. Go for a couple more minutes here. Spongy bone and mineralized portions, right? And so mineralization, of course, is occurring more and more as an individual vertebrate or craniate matures. Very flexible, plasticky, mostly that flexible collagen early on, and spongy bone tissue. And as creatures age and mature, they're going to lay down more layers of hard mineral on top. But this is living tissue, bone cells. And these bone cells are found throughout the bone tissue, and most notably in the marrow in the center, where different types of red blood cells and lymphocytes are made. But there also is blood tissue permeating this bone to bring oxygenation to the marrow. This is an important difference between an endoskeleton, what we think of as a true endoskeleton, versus an exoskeleton. Right? These are cells that are in need of osteocytes, bone cells that are in need of oxygenation and in need of CO2 being removed. So just a distinction between an endoskeleton's two major portions, right? We have the appendicular skeleton on the left, the arms and the legs that hang on the axial skeleton by way of these different types of joints, the shoulder joint and the pelvic joints. And then we see that vertebral column itself, which is quite complicated. An impressive coordinated unit of protection and flexibility. So some stuff you already know here. You may have taken a course of human anatomy or just know this through your years of study. So a closed circulatory system, again, a reliance on red blood cells, which have hemoglobin, that they're the oxygen capturing protein that serve as the way these red blood cells grab and hold on to oxygen to bring to the entire body. And we have a fully separated oxygenated blood and deoxygenated blood system as well. And the blood comes back to the heart and it's pumped to the lungs, comes back to the heart after the lungs, and then it's pumped to the whole body and goes out to all these arteries and arterioles and then capillaries, diffuses across the thin capillary wall, which is only a cell thick, and that gives oxygen to all the cells, and then their CO2 leaves and comes back into that blood. And of course, we don't see blue and red blood. When you look at a vein or artery, the blood color is very similar. You might get a little more purpley, dark red for the oxygenated blood, but you definitely don't get blue blood. That's just a fun. If you see blue veins, that's because the skin's translucent nature causes that color to change to our eyes. And so oxygenation occurs. Here comes the artery. Red blood cells, oxygen diffuses out into the body cells, and CO2 diffuses in, and this is carried back to the heart and then pumped to the lungs again in us and in mammals and in birds, again, to repeat the cycle. Three major blood cells are, of course, red blood cells, RBCs, also called erythrocytes, white blood cells called lymphocytes, which are, they are mostly a protection mechanism for locating, attacking bacteria and viruses, et cetera. And then platelets. Platelets are really important in the blood coagulation cascade. That's the blood clotting cascade where cuts are protected or healed through this remarkable process. Platelets and surround that area, and then a number of other processes take place there. I think we're going to stop right here and just make some interesting distinctions between the circulatory systems of various vertebrate groups. Mammals and birds are lumped together. They have a four-chambered heart, which completely separates oxygenation from deoxygenation. Reptiles have kind of this three-chambered heart that partially allows for mixing of oxygenated blood and deoxygenated blood. Amphibians, three-chambered heart as well, and lots of other sources for O2 coming into the body. These creatures are going to be creatures with lungs, mammals, birds, and reptiles. Some amphibians have lungs. Some amphibians are lungless. Amphibians can also get oxygen into their body through their skin, which is very thin, via osmosis, and through certain patches like the throat. So a frog can simply take in water and hold it in its mouth and get oxygen out of it. That way, inside of its or the skin kind of surrounding its anus is another place where oxygen can get in. And then we have fishes, of course, which have a tiny little two-chambered heart, which we've seen before in dissections, because they have gills, and the gills are a lot fragile, and you don't need a massive pumping mechanism. So a different type of heart that allows for a little bit more mixing of oxygenation and deoxygenated blood. Let's stop there for the day, guys. Have an awesome day. I'm going to post this here in a second, and we'll see you next time.